Good morning. This is webinar 8. This is fungi part 2, pathogen ID. So fungal pathogens part 2, we'll see a lot of the same concepts repeated. We see some different and some same samples. So we're going to just do an overview and really take some time to answer some questions. And I do have a few compare and contrast for you as well today. So anytime anybody has a question or comments, just feel free to jump right in. Unmute yourself, that's totally okay. I'm going to start first sharing my screen, and I do want to show you a bit of a fungal taxonomy, and I promise this is not a complex anything. So did that come up? All right, so this is something that, let's see, is this in the way? I'm sorry, everybody. So, okay, did that help? All right, so this is a bit of fungal taxonomy, and I just kind of want to go through this a little bit with you so you can see the basic subdivisions. This is not technical in any way. I've generalized a lot of things here, but basically fungi are called dicaria. The fungi are divided into two groups, the ascomycetes and the basidiomycetes. So that's where a lot of our conversations happen, and life cycles matter. So remember, in plant pathology, and especially in fungi, we use a lot of terminology, and we look for certain structures as we identify certain structures, sexual and asexual structures, or how we confirm identity. So that's why this matters. We'll start with the basidiomycetes. Your basidiomycetes, or your rusts, your mushrooms, and your conchs and brackets. Rusts, remember, usually have two host life cycles. Your mushrooms, that is a fruiting body. So are your conchs and brackets. So they are fruiting bodies, and that's where spore production occurs. Back up to the top in the red, your ascomycetes, I'm dividing them today into two different groups. Basically your filamentous fungi, those are the ones that have the fungal bodies, the hyphae, and those are what we always consider like the traditional fungi. We also have yeasts. So yeasts usually don't develop that fungal body. They just kind of bud out, and it really resembles kind of a bacterial type style. Your filamentous fungi, though, those are the ones that create, again, that hyphal body as well as some specific fungal structure. So this is really a lot of what we're going to focus on today. Can you see my mouse? The yeasts, besides human yeast and just some saprophytes, the only yeast we really deal much with is peach leaf curl. That is a yeast. Other than that, we're up here in the Pizizomycotina. And within those, we divide again the Discomycota. That's your fungi that produce sexual structure. So they go through a sexual reproduction phase. And the Deuteromycota is a group that we don't see the sexual reproduction stage. Some of those have lost the ability to do so. Some of it we just have not identified in nature. Now, these two groups are no longer valid descriptions. So we use those when we're kind of generalizing and talking. You're not going to find this in the plant pathology literature. This is the old way that we used to describe things. Now, because we have molecular identification, there's no such thing as a Deuteromycota. We put them into other classifications. But just for today's descriptions, the Deuteromycota are the ones that do not have sexual phases. The Discomycota are those that do have sexual structures. So if there is a sexual structure, if there's a sexual phase, the Discomycetes here, we're always going to use the sexual structure for identification, hands down. So usually there will be a sexual and an asexual stage in the Discomycota here, but we always use the sexual stage. That is a rule of thumb. The Deuteromycota, those that we don't see the sexual phase, so in diagnostics we still kind of throw this term around, but the asexual only group, Now we will group with molecular techniques, but if we're doing morphological identification, we still kind of use this. And we consider the hyphomycetes. Those are the pathogens that will create their asexual spores in some kind of structure. Those were our pycnidia, and some of those, I like to call them capsules or survival structures, come in. Or the coelomycetes, those are the naked spores. Those are your molds and mildews where your conidia or your asexual spores are just kind of out there to the environment. So that's how we break these up. I will use some of these terms today. I don't expect you to really know where exactly where these fall. And again, this is just a generalization, but I wanted you to kind of see how fungi are separated. So I'm going to use a few of these terms and I will refer you back to kind of let you know what I'm talking about. All right, I'm going to stop share back here. Does anybody have any questions there? Okay, so let's start with our basidiomycetes. Remember, the basidiomycetes are your rust, your mushrooms, and your conchs and brackets. So we all know, right, the mushrooms. So your mushrooms, they are a fruiting body. Underneath a mushroom, you can have pores or gills, and your spores will come through there. So when we identify mushrooms, a lot of times we'll pop the top off and set them on paper and do spore prints. So mushrooms come in all shapes and sizes. We know that. But identification from the mushroom itself and from the spores doing spore prints is really critical. Different parts of the mushroom are important. So we have a mushroom publication. So if we are going to identify a mushroom, we usually need lots of information, much more than just a picture of the mushroom. So we always need to know if it's a polypore, a gilled mushroom, etc. So one thing about this group of fungi is that they usually live underground or they're wood rotters, and then during rainy weather they'll pop up this fruiting structure, and that's when we can identify. So everybody familiar with the humongous fungus? I think the humongous fungus, which is an Armillaria, the same fungus is connected, I think, across 3 states in the northwest. But anyway, so these guys can be really large, but what you see can only be certain portions. So mushrooms, very much like a mushroom. So some mushrooms and brackets will come out right on the side of a tree or a branch connected here. So you have basically the same thing, but underneath. So here we have gills. So the lines are gills as opposed to just pores, which is the little holes. So much the same thing. They are still within this basidiomycete group. And then conchs, I think this is the turkey tail. This one is the turkey tail, but still connected. These are coming out of a tree. These are usually your wood rotters, and these are usually just harder. Artist conch is common. This one has pores. When I turn on the dino light, I'll show you. So that one has pores as opposed to gills. So anyway, these are pretty common. All shapes and sizes, but usually in our world, an indication of wood rotters. All right, so also within the basidiomycetes, we have the rhizoctonias. Rhizoctonia is a basidiomycete that produces no sexual structures at all, no reproductive structures. It is completely dependent upon the onto the body of the fungus. So I've got some here, a web blight of mum. So we see the gluing, so that the mycelia glue together. When we turn on the dino light, we'll take a look at that, gluing together, and you'll see the mycelia between these dead leaves. Another one, sclerotium. This is your southern blight. So sclerotium, they produce sclerotinia, not sclerotinia. Sclerotinia is a true fungus. They produce sclerotia, which is a fruiting body. We'll look at those as well. So your southern blight or your sclerotium, this is Sclerotium rawlsii that comes from hemp, but it's a soil-borne fungus. So it'll catch right at the soil line, and it'll decay and it'll girdle. see it here where it's girdling. We'll take a look at that. And I have a smaller one here that we can look at. So we'll see a very healthy root system, and we'll see the girdling here. So we'll take a look at some of those structures. And also we have our rust. So a lot of our rust we're pretty familiar with. Cedar apple rust is a very common one. The galls, so remember your rust usually have a 2-host life cycle. So on the rust we have the apple phase, which is on apple leaves, and Isha, which is one of 5 spore types or spore phases coming out, erupting from the back. This has already happened, so the eruption, so these Ishal spores have already infected juniper and cedar for the season. We'll look at some of those under the dino light. This is cedar hawthorn rust. This is on a hawthorn, and this one is in line. So this is common on apple as well, but this one is in line. So a 2-host life cycle there as well. So also on juniper and cedar. So that one is sporulating right now. So I'm going to switch over to my dino light, and we're going to take a look at some of these. So let's see if I don't freeze my computer up again. Share my, where's my dino light? Did it shut down my dino light? No, show all. Okay. Oh my gosh. All right. You should be seeing my dino light screen. Yay. It just gets better. All right. So I've got my dino light here. We're going to start with, well, did you want to look at pores versus gills in mushrooms? Okay, let's look at that so you can. So gills run linear versus your pores. See all those little holes? This is a porous mushroom, or this is actually the conch. See all those little pores? Your spores are going to come out of those. So just real quick, I wanted to show you that. So now let's look at your sclerotium rothschildii. This is your southern blight. Remember last webinar, for those of you who attended the last webinar, we saw sclerotia. Let's see if I can get us on. So there it is girdling. Let's see if I can see my sclerotia. No, it's not cooperating today. We are on a roll, aren't we? Okay, let me get the other one. All right, I'm going to take my dino light off of the stand. Not wearing my glasses. Maybe I'll put those on so that I can see. Here we go. So there are the sclerotia. And if you want to look at them, in terms of size, you'll see the end of my pin. So they're pretty big. You can see those without a dino light. So sclerotia are survival structures. They are basically like a hardened hyphae. So once they melanize, they are ready to overwinter. They can live in soil for many years, and they are resistant to cold, to fungicides, to UV, and to decomposing fungi. There we go. You see you've got different colors, and as they melanize, they get dark and hard. So if you smash them under a compound scope, there's really nothing to see. It's just kind of granular looking. Okay, I know you're all dizzy from that. So this is sclerotia, S-C-L-E-R-O-T-I-A, sclerotia, or a sexual structure, or no, I'm sorry, an asexual structure of of a sclerotium, which is a genus, okay? Another pathogen I was showing you was the Rhizoctonia. Where is, I had a really good leaf earlier. So here is Rhizoctonia. So you are looking at a dead mum leaf. So this is basically, these are two leaves that are kind of stuck together. And so when I pull these apart, or they don't want to come apart, so when you pull these apart, you see fungal body, you see mycelia. Let's see if I can. So they are threaded together. We call this web blight. So they are threaded together. So you see the mycelia under there. And that's a very distinctive characteristic of the web blight. So you see the mycelia. There are no structures at all. There are no asexual or sexual structures. So this pathogen will overwinter in debris, but as hyphae. So this is kind of an odd one. Web blight also affects turf. I've seen it this year on a lot of different hosts, but we see it a ton in mum, those of you who are hort agents. But this is threading together, if you will, of the leaves. So any questions there? Let me get back to my chat so I can see if you are talking. No, I can't see my chat in share mode. Okay, all right. So we can look at some rusts. We'll look at some rust. This is the cedar hawthorn rust. I'm going to go way up. So this is on hawthorn. No, I'm doing a really bad job here. So you'll see the isha that are coming out. So this is a sporulation event, and we'll have infection then of our alternate host. So alternate host and alternative host are two different terms. So, so this is one of our rusts. And what does a rust do? Why do we care? Because we have girdling. No, is that not working? Okay, we have girdling. So this is this lesion is finished, but everything above it, see, is dead because it just girdled it, stopping the movement of water and nutrients to the other end of the branch. And then finally, this is cedar apple rust on apple leaves. This is the top side. You saw a lot of yellowing early in the season, then browning. We get a lot of this at the diagnostic lab. And these are isha. Isha is the structure, the little volcano-like structure. Let's see if I can pull that one off. The volcano-like structure that releases ischal spores. And so kind of a volcano type. That one. Okay, here we go. Let's see if I can focus this. So you'll see on the bottom side, you'll see that release of ischal spores, and that's already happened. So our juniper and our cedar are already infected. So this is a severe infection. Usually with cedar apple rust, our commercial orchards manage it very well. It doesn't take much, but it takes perfect timing to actually manage it. Any questions there on our basidiomycetes? Okay, so we're going to switch back over, and we're going to talk about some of our ascomycetes. I'm sorry, there we go. Our ascomycota, which is your traditional or your more typical fungi. So remember, we have those that produce sexual structures, those that produce asexual structures. Sexual structures will develop early in the spring. So all of your asci will, so we don't have any of those today. At this time of year, we start looking at asexual structures. That's your conidia, etc. So, so let's look at some of those. Remember, we had the those with naked spores and those with enclosed spores. Remember, I kind of showed you that little bit of a diagram. So let's look at some of them with enclosed spores, your coelomycetes. So we've got some really traditional ones. I have a horse chestnut here. We have pycnidia that develop inside. You can see these with the naked eye. So I'll just give you a rundown. This is Phylosticta in maple. We'll see, again, very distinct spots, and with the naked eye, seeing those pycnidia in there. I'll flash all of these through. I have a needle cast of spruce. I have some loose needles in here. We'll show these under the dyno light as well. And we'll see the structures coming through the stomates on those. We've seen those before. And then finally, this is a blueberry tip blight. It's a stress pathogen, but phomopsis coming in, it's a tip blight. And those same structures, those survival structures of some type, this one is pycnidia as well, developing there. So I'll just show you a couple of those. I'm going to do another screen share. Oh, and finally, we're embedding, this is boxwood blight. Kim is embedding these for a program, and I can give you a glimpse, but these produced a sclerotia. So a survival structure from the hyphae, and that's an overwintering structure. So not a sexual structure, but an overwintering structure. So I can show you the symptom there, but we're really not going to see those structures in here. So let's kind of go through those. I am going to share my screen back to my dyno light. And anytime you have a comment or question, feel free to unmute yourself. You can unmute yourself anytime. So this is our maple. So this is a very classic spot of the Phylosticta. You'll see the black dots in the upper left. That is sporulation. Those are the pycnidia. Let me break this off so we can get a better view. There we go. Those are pycnidia at their best. And of course, later in the season, we'll see more production of these type of structures. So if you break these open, they will be like a capsule that is lined with asexual spores. If I can get you closer. They will be full of asexual spores. So notice these types of structures are developing inside the already necrotic tissue. But then look, we've got a border around the lesion, and then we've got that gradient that we've talked about several times. As the fungus, it's moving its way. There we go. That's a really good gradient. You'll see that as the fungus is moving itself away, it's starting to kill the tissue. So it's consuming that tissue, but it's that already spent tissue that the fungus is utilizing to produce those pycnidia, those survival structures. So this is the overwintering phase. So there are asexual spores here. All right, I'm going to switch out, and I'm going to move to our horse chestnut. Let's see if I can find. You can usually see these. Usually see the pycnidia with the naked eye. Let me see what I can see today. There we go. There's some of them. See them in the middle. Lost my pen. You see these in this area. See, they're very light, and so they're just starting to develop. Those right in the center. Let's see if I can get you back. Those here in the center. See, they're starting to turn dark, and they'll turn darker and darker. That's melanin production, and the melanin will protect the pathogen during the winter months. I'll get you down a little bit further so we can look at pycnidia all day long. These are the easy ones for you. So you can see these, you can identify these, and that's how you know exactly that you have a fungal pathogen, you have a fungal leaf spot, and that can be, can you just hit silence, please? Okay. All right, so my phone went off. Unusual, right? So this is a Gignardia on horse chestnut. It's the same pathogen that's basically the same pathogen that's causing black rot in grape. And then I'll show you just one more, and I know you've seen this one before. This is a needle cast in spruce. This is Rhizosperia. You'll see all this is, so we're looking specifically at the underneath of these needles, and they just so happen to all be turned upside down. That's completely luck. This is really severe, okay? So it's not always this clear for you, but once you have a severe case, you'll see the pycnidia are coming right through those stomates. The black are those fungal structures, and each one coming through a stomate. So they have easy access. They don't have to erupt the tissue. They're coming right through. And the little white dots that you see, those are the stomates themselves. So you don't see them coming through. So the white is a healthy, clear stomate, and the black is the actual structure coming through. So they usually line up kind of like tin soldiers. And again, this is extreme, but if you're looking, any of you who are working with needle casts and you call me, if you call me or ask me, what do I look for, how do I sample, this is what I'm going to send you after. And this is the older, more severely infected needles. So these structures are developing on needles that have been infected for a while. Okay, so they're not going to develop right away. So first you'll start seeing maybe some yellowing, some casting, and then later on you'll see the production of the structure. So sometimes we have to go to the fallen needles on the ground. Even if we're looking at some of the, like the Diplodia, tip-blighted pine, we're looking at those fallen needles or the older needles, those on the ground, or even the pine cones on the ground. Any questions there? Oh, I wanted to show you the box with light. So any questions on the coelomycetes? Wow, we've got a glare. There is someone that has called my office phone, now my cell phone, and sent me an email. So this person is really persistent. Okay, so what I'm attempting to show you without the glare. Kim is digging at the bottom of my bag looking for my phone. Okay, just press any. Okay, sorry about that, everybody. But anyway, so this is the little purple lesions that we're trying to capture, and we've embedded these. See here, we've embedded these because we don't want boxwood blight escaping at any of our events. So, okay, there's a glare. I will bring these to the practicum if anybody wants to see them. So it's just not really translating well. So you see the purple lesions, and then sclerotia will develop at the end of the season. And remember, the sclerotia does not have spores inside. That is the hardened mycelia, and they create those little kind of like little balls of knotted hyphae. Okay, do we have any questions there? I'm going to switch back so I can see the chat box. All right, any questions? All right, that was coelomycetes. Coelomycetes are your ascomycota that produce some type of structure or capsule to host their asexual spores. All right, now we're going into the hyphomycetes. Hyphomycetes are your naked spores. Those are the molds and the mildews that we commonly get. Oh, I just dumped all of the roots. Okay, so with those, we can induce sporulation. Remember, we've talked about moist chambers, takeout containers, Ziploc bags. So a lot of times you can induce sporulation with your moist chambers. So with your hyphomycetes or your naked spores, you want to see those molds and mildews. So let's see, where am I going to start? Oh, how about a really common one that people mistake? I'm going to show this one. Cheesy but true. Sooty mold. See the sooty mold going on here? There it is. Sooty mold is actually a mold. These are this is a fungus that's growing on the sugars that are leached by insects, sucking insects. So basically feeding on the sugars in insect poo. So sooty molds this is the one that I always refer to the entomologist, and not to weed plant pathologist. So it is a fungus, it is a mold, if you will, but it is It is a result of, it's a secondary. So this fungus is not infecting or affecting the plant. It's the insect, whatever it is that's sucking. All right, so next, another, your very classic hyphomycete is, guess what? Powdery mildew. Powdery mildew is very classic, white and powdery. Let's see, let me give you this one. This is what happens when the plant pathologist does not spray her garden. So this is solid white. Powdery mildews typically infect the top side of leaves. Remember, that's opposite of downy mildews that are not UV tolerant, and they'll usually develop on the bottom sides of leaves. Powdery mildews like arid conditions, so it likes much less of the humidity and water than the typical fungi do. It needs a humidity, but when it's really rainy, we usually don't have a whole lot of powdery mildew. Powdery will start kind of spotty. You can't see spotty. We'll switch out to my dino light. It starts spotty. This one in particular is kind of on the back side of the leaf. And then I will give you some close-ups. This is a bee balm. I've cut it. This is bee balm in my yard. You'll see the powdery mildew. So we'll switch out to that. And also, so we're calling them and we're classifying them as hyphomycetes, but we do get sexual structures in powdery mildew. So Chasmathecia, and I'll show you those are black dots. So I'm going to switch out to that in a second. So this is one that is typically those naked spores that we're so familiar with when we talk about the words mold or mildew. Powdery mildew is that classic example. Some others that you may be familiar with is Septoria. Okay, I had a Septoria leaf spot in hemp that was somewhere on this bench. Septorias, they produce, this is a tomato, little small dots inside those. We'll see the spores that come out, the conidial spores that come out. They kind of look like octopus legs. They're white and they just kind of ooze out like in a curling motion. So that's how we identify those. And then I'll also switch out and show you some Cercospora and a new fungus that we have on hemp. And I'll show you just kind of some of the challenges that we're kind of going through with a new fungus. So I'm switching back out to a share screen. Any questions? If you're putting them in the chat box, I can see them now. Once I share my screen, I can't. So if Kim doesn't see them, just unmute yourself and ask questions. All right. I'm supposed to be sharing my screen. There we go. All right, there's my share screen. So let's talk about powdery mildew. First, we all know the powdery mildew classic examples, but powdery mildew produce chains of spores, one conidia hooked to another, and that's what makes them fluffy. Okay, so I have just tortured this one to death. So powdery mildews, this is what you would see in a hand lens or with your dino light. See if I can get closer. So you'll see the hyphal mat. And then some of these, I know what I'm looking at. I guess it's not quite so clear to you. The chains of spores that are developing here, and that's where the height really comes from. I can get you a better image. You'll see some of the leaf structure. You know, we always talk about knowing what the leaf looks like without the fungus. So we see leaf hairs that are pretty prominent here in this bee balm. So some of these, you see the chains of spores. Let's see if I can go. See here, the chains of spores. See that? Those are chains of spores. And I wish I could zoom it in, but I am not. Let's see. Circle. Yeah. So those are chains of spores. So those are some examples. And if this were your light, or if you had it in front of you, you would see it much more clearly. Okay. Let me delete that circle. So I can switch out to maybe, just maybe, we'll get some clear chains here. on the squash. Okay, those are larger spores, and you'll kind of see those chains going on here. So clear chains. There's some more chains of spores. Okay, does that make sense? Okay, and this is really severe. So, all right, I'll switch. This is an oak powdery mildew, and I'll show you again the chasmathesia. This is the overwintering phase of the powdery mildew. These chasmathesia are have appendages, so they're like Velcro. They will stick, and you'll see the one on the bottom of the screen here. You'll see these kind of appendages that stick out like a starburst. Sometimes they're hooked like Velcro, sometimes they're not. So this is the overwintering phase, and they will stick. They will stick to anything they please. They will stick to wood. They will stick to bark. They will stick to fence posts. That's one thing that grape growers struggle with, is all the different surfaces that powdery mildew attach to. So on the bottom left, you'll see more appendages. They're a little bit clearer here. I hope they're showing up well for you. Okay, let's see if this guy will give us a good view. There we go. You'll see those appendages coming out. So three-dimensional appendages. There we go. So this is the overwintering structure. Notice that they're melanized and dark. That means that they are tough and they're ready to survive the elements. Questions there? All right, I'm going to pick up and show you some help. Here are a couple of different, oh, let's see, let me switch to, let's see if I can get a septoria. I'm going to show you the oozing, or I even marked this one for you. I marked this one, so hopefully we can get this one pretty quickly. See that? Okay, so I made a square with a Sharpie, and then in the center is a septoria spot. So septoria is doesn't really have that distinct center, not the necrotic center that we're used to. Okay, so septoria is basically black across the board. Let's see if I can get this view. Darn it. There it is. There it is. Let's see if I can set down my. Okay. Maybe too bold. Okay, it's not showing up here. So there are just some octopus-like, so the white you see in the center, we're not getting. So the white you see in the center are spores. These have kind of just collapsed after being in the bag. Let's see if I can find one more, and then we're not gonna spend a ton of time on it, but they're really worth seeing. They're really cool. But that's the white you see in such a lesion. Is the let's see, try. No, I miss it. Sometimes this seems so easy, and then it just is not. Okay, well, I'm not going to waste too much time. We're going to see some of this stuff at the practicum, where it's a little bit easier for you to do it yourself, and not have to worry about someone else in a screen. Okay, that's just the hole that fell out. You'll see on this area. So, oh, sorry. What happened? So anyway, this area. you'll see the sporulation. But we'll do it at the practicum so you can see a little bit better. Uh-oh. What did I do? I'm so sorry. Why did I lose the size of my screen? Where's my little, did you put in the corner? Should I not there? No. Today is just not my day. I don't know what the heck. Okay. Okay. Oh, I just took a picture. Okay. Nope. No. Click something, and as usual, I made a mess. Okay. All right, so we'll just have to move on, and I'm so sorry. I want to show you a couple of different diseases. This is industrial hemp. Love how we say the word industrial, right? So this is hemp, and this is Cercospora. So those of you who work in other Cercosporas, whether it's in tobacco or in soybean or in any other system, Cercosporas are classic. They've got a very distinct purple border, tan center, yellow halo, and I want to compare that to a new disease that we are working with. We're calling this one Turgeonia, hemp leaf spot. This one is really common, and you'll see it is lighter. See, the small one is a Turgeonia, and as it gets larger, it doesn't have that purple border as much, and it doesn't have the yellow halo. And then we will see coalescence, and this is a term that is important for you all to know. So as spots meet up with each other and kind of combine, we call that coalesce. So there's the coalescing of some lesions, and then what becomes almost blighting, so it's the blackening. So I did want to show you that. And I will switch over. I'm going to show you some pictures in a PowerPoint just to give you a really quick overview. Any questions, by the way? Making sure nobody has questions. All right, so I am going to share. So share, here's some leaf spots. Okay, so this is a Cercospora leaf spot. What I did some close-ups here. This is going to be part of something else, but I did want to show you. So this is exactly what I just showed you. You see the yellow halos, very clear yellow halo here on the right. But look at the center. See the conidiophores, and conidiophore are the little stalks that hold up conidia. But you see they're clustered. There are several of them, about 7, that come out of a single area. They come out together as little clusters. So as we start diagnosing, we need to see all of these things. The white in the centers are usually just dead tissue, and that's usually where we see these clusters come out. Just the opposite here is a septoria leaf spot. So we see a little bit of yellowing with septoria, but overall septoria are uniformly the same color. They are more angular. They start as black specks. See right here? There's kind of more of a black speck. So it doesn't start with the yellowing. Here we go. There's a black speck in the center. And these are those curly octopus type conidia that are coming out. So they're curly. And that's what makes the white center. So the tissue never becomes white. And then there are scattered pycnidia. You see the pycnidia? That's pretty common with septoria. We see pycnidia, which are the survival structures. Inside will be asexual spores. And in the right climate, that's when those pycnidia can also germinate. So you can have these white conidia coming out of the tissue if the climate's right, or coming out of conidia if you have a dry spell and followed by a wet spell. And this is the hemp leaf spot that I was just showing you. You see kind of a more narrow border. Symptoms will manifest differently, but here are the conidiophores all the way to the right. The conidiophores are scattered, so they're not clustered like in Cercospora. So you don't have to memorize this. I just kind of wanted to show you that we have very similar leaf spots from the surface, but drilling down and knowing some of the details is really important. And that's a lot of what we do, and that's what our diagnosticians do as they work through some of these things. So of course, it's hard to explain to our clientele, to our stakeholders, exactly what we are aiming for and why we ask so many questions, but this is why it's important. The questions there. All right, stop sharing my screen. Any questions? Okay, so just a couple more minutes, we'll get through. I have some downy mildew, if anybody wants to compare, contrast downy and powdery mildew. I also have a fungal root rot that we can compare to a pythium, like an oomycete root rot. So if anybody is interested in that, I'm going to go through that right now. And I am going to share my screen again, and let's see if I can clear up my issue with my DynoLite screen. There's my DynoLite screen. Again, I have somehow minimized the size. I did something, and I just don't know what. Settings, is that going to give me an option? Back to this. Yeah, that's the largest. Okay. If anybody wants pictures of any of this, let us know and we'll screenshot it and let you see. So fungal root rot, remember this was the Leviopsis, black root rot. Oh, my light's even messed up here. The Leviopsis is really, again, it's a fungal root rot. Let's see if I can go down here. It comes in from the side, from root hairs. So we'll see lesions coming in from the side. Pretty common that we can identify. You'll see a white root. I moved it. You'll see a white root, and then you will see the black midway. So you see white, then black, then white. Let's see if I can move up a little bit. White, then black, then white. You see that, those black lesions. So that infection is coming in from the side. Pythium and Phytophthora, which are your oomycetes, are much the opposite. They are going to infect from the tips of the roots, and Pythium in particular, this is a Pythium, it causes what we call rat tail. That's the sloughing off of the cortex of the root, and then you're left with just the steel. So you'll see just the steel hanging out of these. So that's very classic of your Pythiums and your oomycete root rots. And a lot of times, here we go, this is a pretty classic example. And you'll see here is all of the steel, and then right where this soil is intact on that cortex. And if you hold those with your hand, you can usually slough off that that cortex really easily, or even as you're pulling roots out of a soil. So if we're going to look at these from a diagnostic standpoint, we really want you to leave all the soil intact. And that kind of helps us take a look at it. We can look at EC and pH. Wrong way. Okay. So again, we call this a rat tail. So the steel is all that's left on the end. Any questions there? All right. And then I'm going to just really quickly show you, remember your powdery mildew, remember your chains of spores that we saw. Let's see if we can capture some really quick again. So your chains of spores, and oh wow, here's a really good one. right here. See this chain of spores? Let's see if I can make a circle. Well, there's 2 chains of spores there. Will that, nope, that doesn't do it. Okay. I know when I try to move this thing, it will, no, it's not cooperating. So chains of spores. All right, so let me switch out. Delete this square, okay. And I have downy mildew. The first time I've seen downy mildew in impatiens this year, not that it matters what we're looking at it on. Downy mildew is very typical on the undersides of leaves, not the upper side. We have a focus here. There we go. So downy mildew, even though it's on the underside of leaves, you see a lot of yellowing on the top sides of leaves. So defoliation is what kind of triggered this for me. So I'm going to pull off a leaf or 2, and we'll see if we can see some of the classic downy mildew. So who remembers what we learned last time when we studied the oomycetes? What are the distinguishing characteristics of a downy mildew? Does anybody remember that? What's that I'm going to get out of it? Let's see if we can get a little bit more. Okay, so, okay, well, I guess we're not going to get, so you see the clusters, these little clusters, the little branching. So those water droplets, those are water droplets from a humid bag that I had it in, but all around here. This is the clusters of your spores. Now remember the antlers, we talked about antlers. So we would have had to do this on a, with a slide. We will definitely do that at the practicum. So the antlers, this is the underside of a leaf and more clustered, whereas the powdery mildew, you can see the chains of spores much more clearly. Sorry about that. I was sure we'd be able to see it. I was absolutely sure. So we see some raised okay. So we're not a happy leaf today. So powdery, downy mildew, I'm sorry. Downy mildew. Underside of the leaf. And this is what I showed you today is pretty much all you'd see with a hand lens and having to go in later. You see a lot of downy leaf hairs too. So knowing the difference and being able to see the difference going on. Any questions there? All right. All right, so I guess you can't see what I wanted you to see. All right, I'm going to stop sharing, and that is about all we have today. Last chance for questions on that. All right. So, Kim, have I opened the quiz? I haven't opened that email, have I? Okay, guys, just let me open that email. Kim and I send each other emails, and that's how we move files sometimes when we're real time. No, that's not right. All right. There we go. All right, let me share my screen with you now. All right, training website is where you're going to go. You're going to find this video in about an hour. You can always go there. That's where you will also find, so if you listen to the video all the way to the end, that's where you're going to find the link for the quiz. All right. Those of you who want the link for the quiz, this is going to be it, and Kim has put it in the chat box for you. Just double check. Kim has put it in the chat box for you. You can click that right now and it'll take you directly to it. So I am going to switch back over for a minute and then I'm going to leave this up for those who are watching the video later. Again, questions? So those of you who are on, thinking about the practicum coming up, what are you expecting to practice during the practicum? Any thoughts about that? Do you want to do more canker and woody tissue? Do you want to do more root tissue? Do you want to do more close-up and compound scope kind of thing? Oh, Kathy, if you want CEUs, you can take the quiz again, and you'll get an hour and 15 minutes of credit. If you've already taken the webinar 3 quiz, then that counts for the certificate program. But you can absolutely get credit for today because it was different. So we are going to turn this in for your CEUs. So if you want to take the quiz again, that's fine. So those who get credit are only the ones who complete the quiz and you enter your name. That name, when you enter your name, it generates it onto a spreadsheet, and that's what we hand over. Anything you'd like to do specifically in practicum? Those of you who are on, you can put it in the chat box or unmute yourself. No? Because, so some of our plans, we're going to do a field walk. Okay, some things with roots. Woody roots. Okay, cankers. All right. So we're going to do a field walk, and that's just going to be how to look generally and how to consider everything, how to take the information. And we're going to come in and we're going to do some hands-on. I'm going to let you stream that bacteria with me looking over your shoulder. I'm going to let you, if you want to crush some pycnidia and see the spores inside, I'll try to get some root knot nematodes and let you see the eggs inside. So those kinds of things. So we can cut some things open and look at some things more closely. Then during lunch, we're going to have a working lunch. And that's because I've got to have you for 8 hours working. So we're going to have a working lunch and we're going to just talk about management. So we're going to give you some diagnostic forms with a diagnosis, and we're going to let you kind of discuss in small groups while you eat some management options. How would you handle management on a commercial field versus if it was a homeowner versus if it was a valuable plant. So we're going to do some of that stuff informally, but we still are going to have a working lunch. We're providing lunch for you. It's sandwiches in all locations, box lunches. We're going to have some choices. Then at about 1 or 1:30, we're going to have some case studies for you. We've been working on case studies. We're having some problems with a few inoculations, but we will keep you busy for 2 hours. So our goal is 4 case studies, 30 minutes a piece, so 2 hours. That takes us right into the 3 o'clock mark. And we have to keep you until 4. So we're going to survey the heck out of you. So any kind of question and answers, anything you want to go back over, any learning there. And then, of course, just going through, we have got to capture some information here. I know we've already surveyed you on webinar 7. We gave you a lot of, boxwood blight. I will bring some boxwood blight. So we surveyed you on the webinar series. We're going to take a look at that. I know we had a lot of computer glitches. So if we do this again in 2 years, we can really improve the computer glitches. But we've asked you a lot of questions. We're going to ask you questions at the practicum about the practicum in particular, what it is you want to see, what it is you want to do, and how you can apply that. And then those who completed the certificate program, we will survey again, probably in a week after that. And we're going to ask you about the program as a whole. And we're going to try to capture some information. So it's not about giving us lots of compliments as much as helping us to do this again, Do we do annual trainings just to get you some CEUs? Maybe we can pick certain topics through the year, maybe quarterly. If those are things you wanted to do to kind of update yourself on what's going on, or some techniques or whatever. So those are kind of what's in my plan coming up, especially for the practicum. Any other questions? So I'm going to bring lots of cankers and lots of woody roots. Their college textbook, we can get some of this on. You know, unfortunately, some of these textbooks are just really plant path driven. I wish there was something, but if you really want to read into it, there's some really great books that we consider great books. But as for you to read, unfortunately, I don't know of any. I will look, though, and I will bring some resources in. Oh, I have another sample here. I have some senescence, and we didn't go over that today. Just what happens when some samples come in and plants are starting to senesce, and all of the different kinds of spots and scorches and all the symptoms that come in with yellowing and kind of spent leaves. So that's something else I didn't do today that I wanted to kind of show you, but I'm going to let you go because we're almost at time. All right, so Kim is still giving you the link. I'm going to share screen again so that those who are watching this recording see the link. This is the link to the quiz. Anybody who wants credit for this 1 hour and 15 minutes CEU, this is the link you go to, and you're going to put your name in the box. Those of you who are watching right now or live right now could just click the link in the chat box. All right, unmute yourself if you have any last minute questions, or if you think of something that you would like to do or see during the practicum, just let me know. All right, so those of you who are on, you are free to go. You can escape. And I promise not to have another webinar until at least next spring. This has been, I know this has been kind of frustrating, some of it, but then at the same time, we're trying to keep you in your seat in your office instead of traveling. Hope to see you all in September. So we have four practicum trainings coming up. So looking forward to seeing all of you at one of those 4. And never hesitate to contact us. Kim or I are always here to help you out. So have a good weekend. Have a good holiday weekend, as a matter of fact.
You too.
And we're escaping out of here. It's the first home football game tomorrow. So all the tents are already coming out on campus. And the motorhomes park in our parking lot. So we get kicked out anyway. So we're going to escape as soon as we can today as well. So have a good weekend. And I will see you all in September. Thanks.